Today, I'd like to tell you about a technology that has the potential to be the next stage of human evolution. This technology can cure diseases and genetic defects, improve crops, and slow aging, but it does raise a few ethical concerns. That technology is CRISPR. No, not your vegetable drawer. CRISPR, short for CRISPR-Cas9, is a tool for editing genomes that scientists have developed from bacteria. CRISPR as a concept has been around since the late 1980s, and in 2013 the technology was first used to edit human cells. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and this gives us an idea of how it works. DNA is made up of a series of base pairs that determine all of our characteristics. Errors in the sequence can cause a whole myriad of health problems, and this is where CRISPR comes in. CRISPR works by editing the repeating pairs of an organism's DNA. It targets specific sets of genetic code and edits the DNA at that location, permanently modifying the cells of the organism. With CRISPR, DNA pairs can be replaced or removed altogether. In humans, genetic diseases such as autism and muscular dystrophy can be removed. Cancer and viruses like HIV can be eliminated. Research has also shown that CRISPR can be used to slow down the aging process. CRISPR can also be used to improve crops or eliminate species that are potentially harmful to humans. Used on adults, CRISPR only works on an individual basis. Typically, these modifications would die with the individual. The exception to this is when CRISPR is used on reproductive cells or very early embryos. Used in this way, gene edits can eventually spread through the population, potentially removing a variety of genetic defects. CRISPR is not only limited to genetic defects, it can also be used to make improvements to human DNA. This is where ethical concerns start to arise. Modified humans can be made that are stronger, faster, smarter, and better looking. Science will have to decide where the line exists between ethical and unethical when modifying future generations of humans. CRISPR is of interest to me because I have a genetic defect that is so far only curable by technology like CRISPR. I'm colorblind. Specifically, I have red-green color blindness, which is known as deuteranopia. The retina of the eye has two light-sensitive cells called rods and cones. The cones in the retina are responsible for determining color, and my color blindness is caused by a genetic defect in the cone cells. A company called NeedsLab in Washington is working on a fix. They have currently used CRISPR successfully in monkeys to cure color blindness, and here we see a clip of a CRISPR-treated monkey taking and passing a color blindness test he was not able to complete before gene therapy. NeedsLab is currently awaiting FDA approval for human testing. CRISPR is a powerful tool that has the potential to change life as we know it, as long as we give it the respect it deserves. With careful research and implementation, humans everywhere can greatly benefit from this technology in the years to come. Thank you.